Can you put on mics when you get a second?
Oh, yeah. Yeah. I think so. All right, my name is Hayden. For the last 5 years, I've been working at a cardiac MRI software company. And what we have been basically building is software that looks at our pulse sync, which is called fast sync. And so what it is called a strain encoded sequence. What you're looking at is you're looking at multiple cross sections of the heart, and you're seeing how the tissue actually contracts. So you're sending two different frequencies, low tune and high tune, and what you're able to measure from that is actually how much the tissue itself is contracting. So we would build deep learning models from scratch on tens of thousands of our images to actually predict where the heart is on there, and then we would actually quantify from those segmentations how the heart is performing on a regional basis. But one of the downsides is it only works on fast sync itself. It doesn't work on any other cardiac modalities, and I would have to get a lot of training data to try to extend it beyond that. And so that's where I started building this web app called Radiology Lab. And it is extended to cine data, it's extended to brain MRI data, it's extended to cardiac CT data, and I have it integrated with what's called SAM3. SAM3 is a very popular segmentation model. It's a transformer-based model, so it's a little bit heavier. And here, I'll load a data set right here. What this takes in is a DICOM data set, which is a medical imaging files format that is very popular. And once this loads, you'll see right here that I have what's called a volumetric renderer. It's basically a ray marching based shader, all written inside React 3. And this is a cine heart modality. And so there's a bunch of different pulse sequence that you can look at, which all show different representations of the same data. So this is one scan for one patient and multiple different heart modalities. And this is the pulse sequence at the company that I worked at. This is fast sync. So when you press play, you can actually see it change colors. And what that represents is the actual tissue contracting itself. Well, on cine data, you also have the motion, but it's much higher resolution. And our fast sync models couldn't actually predict on this cine data. And so I set up a API on Hugging Face for SAM3, and I'll enter the key, connect it. And the nice thing about SAM3 is it allows for a lot of different types of prompts. So it doesn't just take image in, segmentation out, you actually have to guide it. So you can either show it a shape or you can actually type what it's supposed to draw. And so I'll search right here for myocardium, which is actually like the tissue in the heart. And after a little bit, it's a pretty heavy model, you'll see that it's actually contouring the myocardium right here. And you can do this for multiple planes, which you can see on the right here, and add to your data set. But if you look on this on a 2D basis, you'll notice that it's kind of drawing stuff right at the top of the heart. And that's probably not something that I would want the model to actually draw. So what you'll do is you'll click on the object and you'll actually try to erase that section, accept your changes, and run it. And you'll see that it actually adjusts all the images throughout. The nice thing about this transformer-based architecture, it's able to use context throughout and updates its history. So one other data set that I wanted to show was, so yeah, Yeah, so the objective is to help people label data so they can build training data to train other AI models. And then they can hook those AI models into the same platform, and then it will be geared and fine-tuned to their heart modalities or their brain MRI, whatever their imaging is. So reloading this, I'll open a brain MRI data set, which typically has a lot more planes and a little bit higher of a resolution.
So this data which you have, is this for healthy volunteers or is it like just subjects?
Sorry, can you repeat that?
So are these healthy volunteers or is this like actual data?
This is actual data, yes. So this data set was open sourced through the Cancer Archive online, the Cancer Imaging Archive. And so I just downloaded that. And as you can see, this is called a flare imaging modality. And you can kind of see these bright regions, which would be anything from inflammation or edema or something along those lines, but they're unhealthy regions. And I'll rehook the SAM3, and what I'll do for this one is I can't just describe this object. It's a little bit more difficult to describe this object. So I'll actually draw it an example first. So I'll create a new object. I'll paint this object all in that red region that I want it to propagate through time. And so you'll notice there's a little bit of holes and whatnot. And I'll accept this. I'll change it to run for the entire view, the entire group of planes. And then you'll see it from the side. You'll see just one plane right here. But then you'll see it propagated that through the entire view. Since there isn't actually multiple frames for each of these planes, you can just see it throughout. Now that I have these segmentations at every single plane across this, what I did is I actually hooked up what Ron was describing, Gaussian splatting. And so now that I have all of these segmentations, I actually want to try to create a mesh around it. And sometimes you'll have planes that are very sparse. They're very far apart. And what you actually want to do is you want to kind of fill in the gaps. And so what I used is the gsplat library. It's a Python library that actually is PyTorch kind of based. And it actually trains to the images themselves. And when I say that is it will actually fit and predict everything that is within inside this mask itself. So it's predicting the images and the mask at the same time. Then what happens is you get that entire volume and then you predict a mesh that encloses that using what's called ray marching. And I'll hook up my Gaussian splatting API as well. And you'll see as I...
Is the training because there's a need from the operational standpoint, in other words, people operating, or is it because they're diagnosing, or is it
So,
there, or what's the need for the estimation aspect?
Yep. So as you are kind of trying to, I'll say it for the company that I worked at, is we wanted to identify where the heart is. So if I had this platform and I could draw a bunch of examples where the heart is, I could take the big model and fine tune it so that way it will learn that representation. So this is just to help people build training data on a more general basis. It won't just work on one imaging modality, it will work on any sort of DICOM format. But yes. But this specifically is you're actually training to predict where Gaussians are in the actual brain. And so when I switch over to the mesh, you'll see just the mesh itself. And right now it is trying to fit a bunch of ellipsoids to represent the actual underlying bright intensity values that I segmented. And so as you can see right here, you actually have a object that is a lot more higher fidelity, and you actually have a mesh that can then be exported for, let's say you have a neurosurgeon that wants to actually quantify how big a tumor is. They're able to use that. Well, you also have the data that's inside of it as well. All the Gaussians, all the intensities, all the different fields that you can export as well. So the next step is to take this mesh and what's called tetrahedralize it, basically just make it a volumetric mesh, not just stuff on the outside, but stuff on the inside. So that way researchers can actually model it in whether they're finite element kind of analysis software and whatnot. Anyways, this is my demo, my project. Mhm.
Sem3 is kind of a new model. Have you tried it with other segmentations before? Or
No, so I actually started this in early December, building this web app. And so SAM3 was, exactly, and it's all open source, and it's nice to be able to describe what you're supposed to draw. I think one thing that I forgot to announce is like, when it comes to medical imaging, SAM3 is trained on data from everything. It's not fine-tuned to be very accurate on medical images. That's just a very small amount of what it's trained on. So when you try to describe something in a medical imaging kind of context, it's not going to perform that well. If I just say tumor, it's not going to know where that is. Maybe it will if it's trained on it. So the idea is you need to create training data and then hook it back into this and then kind of iterate.
So you're using Dicom. Dicom is super difficult to use. It's a heavy
Yeah.
file.
Yeah.
How are you running SAM3? Are you slicing and you are doing like a video
So-
for the visualization and also for the processing? How you are managing this Dicom format? And
Yeah.
what is the infrastructure that you have behind this web browser?
So this is a React web app. So it's all JavaScript. And as far as how it's extracting it, it's using a library called DCM.js. It's just a way to, on the client side, parse all the different dicoms and extract the images, and also important information like the position of the plane itself. So you'll see right here, it's extracting all of this image data, and then it's extracting a lot of different information about each of these planes, and then plotting it all in 3D space in a volumetric shader.
Regarding the infrastructure, are you using, if this is running in your local machine, or you are using a GPU or a data center?
So for the APIs, the SAM3 API and the Gaussian splatting, I am using Hugging Face. So I'm just deploying it through that. But for this, this is all just locally, but because it's in React, I can host this online if I would want. I've noticed a lot of hospitals today are kind of leading towards a kind of server infrastructure, just a private URL that they can access. Mm-hmm.
So your intent will be to sell this eventually if you create, like, you develop this to a hospital setting where a cardiologist can use it on his different scans, the real-time data, or maybe for spleen measurements, I'm not sure if it measures the spleen, but it
Yeah.
does that if, and real-time data has been fed into it.
Ye-
Is that what the intent is?
I think the intent is to help people build kind of machine learning models to automate segmenting their medical images. So yes, the intent would be for hospitals or researchers to use a platform like this, and then to build their own models that they then have to validate. If they were going to use in a clinical setting, you would have to get it FDA approved. There's a lot of hurdles beyond that. Mm-hmm.
So the output from this process is another file basically of the same format?
So if I were to export it, I would export all of the images that were extracted. So a popular format and a popular data set is called COCO. So I would probably export all of the images, any other important information, so that way it can be easily fed in through something like RoboFlow to actually fine tune a SAM3 model. And then you would hook it back in.
And the to fine-tune SAM3 model. SAM3 is basically a base
Yep.
foundation model, so this allows you to tune it for that specific
Exactly.
scenario.
Exactly. I don't have a separate web app to help you with fine tuning that. So yeah, you would export the data and you'd have to manually fine tune it. And as long as it satisfies the API contract and can communicate with it correctly, then you would just hook it back in. Mhm. Yep. Thank you.
And also the best demo award.
Oh yeah.
Yeah.
This way.